Charles, the cell, mitosis. Meet Charles. Currently, he's engaged in metabolic activity and getting ready for the first phase of mitosis. Before mitosis occurs, the cell goes through 3 phases which are collectively termed interphase. Inter means between. The interphase is the composite phases of growth and synthesis which occur between mitosis and the first growth phase, or G1. Charles is now entering into the first phase of mitosis, called prophase. As you can see, the nucleolus is no longer visible and Charles's chromosomes are beginning to condense. We are already starting to see some chromosomes. The centrioles have split up as well and are beginning to migrate to opposite ends of the cell. Charles's centrioles have made it to opposite sides of the nucleus, and we can now see the spindle fibers of each centriole are formed. What's missing here, though? Looks like pro-metaphase has started because the nuclear envelope is gone. Metaphase is about to begin as the chromosomes are moved to the center of the cell. It didn't take Charles very long to get those chromosomes in place. Quite the orderly fellow. Metaphase is complete once Charles has all the chromosomes along what is called the metaphase plate. This organization ensures that when Charles splits, there will be an equal number of chromosomes in each cell. Anaphase is now beginning, with the spindle fibers pulling at the centers of the chromosomes where a small protein structure is located called the kinetochore. The spindle fibers attach to these kinetochores and pull the chromosome pairs apart. The centrioles also move farther apart as the chromosomes are separated and taken to opposite poles. Charles is almost done separating himself into 2 parts. He'll soon have a split personality. His next step before that can occur, though, is telophase. We can see here in telophase that the chromosomes are becoming less recognizable. The spindle fibers are pulling back. Charles is almost finished as the chromosomes decondense and 2 nuclear envelopes are formed. As you can see, there are protein fibers forming in the middle of the cell. This is where cytokinesis begins. Cytokinesis is the process of Charles splitting into 2 cells. In animal cells, like Charles, the fibers pinch the cells off from each other. In a plant cell, a cell wall simply forms and separates the 2 cells. I am proud to now introduce you to Charles II. And of course, you know Charles I. Charles just went through the process of mitosis where he started at prophase, moved on to metaphase, switched to anaphase, began to finish up at telophase, and completed separating from himself with cytokinesis. Charles I and Charles II will have to go through their growth phase now, but after the 3 stages of interphase, both Charleses will have to start mitosis again. I wonder if they ever lose track of how many copies of themselves they have.